Let me know in the chat box. Am I audible? We are having the class with the topic of immunology and serology. Are you uh, are you aware with that? What is immunology and what is serology? Do you heard this term? Can anyone tell me about the immunology? What is the meaning of immunology and what is the meaning of serology? Yes, can anyone? Okay, so the immunology as study of immune system, right? Immuno means your immune system. How your immune system work, what are the different components that come under immune system, and what are the different organs of immune system is all covered under immunology, right? So the study of immune system is immunology and the study of serum is known as serology, right? So let's start with that. I'm going to share my screen. Yes, this is visible to all of you. My screen is visible to all of you. Oh, all right. Okay, got it. All right. So start with the immunology and serology. So I already told you about the immunology is the meaning of is the word of immunity which derived from the immunity. So basically we will discuss about the immunity, how our immune system work and what is the immunity and how it is work for us. So this is the study of immune system. So this come under immunology and the serology, the study of serum. In our serum antibodies are present, so we will discuss about antibodies, how we can detect antibodies for different diseases for different by different methods, right? So this all come under again serology. So let's start. So after the end of the class you all will be able to answer what is immunology, type of immunity, types of vaccine, serology, serological reaction. So in our class we will discuss about these in detail, right? So first let's start with the immunity, right? So what is immunity? Immunity basically is a condition where individual protect ourselves from the diseases. So our immune system protect ourselves from the diseases is termed under as immune system, and the study of that immune system come under immunology. So in simple words you can say immunity to disease. Here what is the difference that let's suppose a person is come with the exposure of typhoid antigen, right? So his or her body start producing antibodies against that antigen and provide immunity to fight against that antigen. So this scenario come under immunology. So basically immunity is generally two types, innate or acquired. Innate is initially, which provide immunity initially by birth, come under innate immunity, right? And acquired, which we acquire after the birth by our environment, by different vaccines, by lifestyles. In whatever way we acquired anything or acquired an immunity, come under acquired immunity. So further there are acquired or active immunity having further divisions. If I talk about acquired immunity, it classified under active immunity and passive immunity, correct? Active immunity again is of your naturally acquired, artificially acquired. Passive immunity, naturally acquired, artificially acquired. So this is the subdivision of acquired immunity, correct? Now see in this image. Active immunity. There are 4 types of acquired immunity. If I talk about active acquired immunity, so in active there is natural. A person naturally arises or naturally produces antibodies by the exposure of antigen. As I already gave you example that a person is come in the exposure of Vidal antigen and our body start producing antibodies against that. If we cope up with that disease without taking any artificial or any medicine, then it come under natural, because our body produce antibodies by herself to fight against that disease, right? This is the natural active acquired immunity. Now artificially acquired active immunities are those which give outside the body. So artificially acquired active immunity results from an inoculation of toxicoids or vaccines. Artificial immunity get by vaccinations, right? Now the passive immunity. If I talk about natural, so it naturally pass from mother to fetus, right? By the help of placenta. So IgG is a type of antibody which is passed through mother to fetus with the help of the umbilical cord. Now artificial. Artificial acquired passive immunity is induced by an injections, right, of antibodies, which is taken from the circulation of an animal or other person. So passive artificial immunity is the one which is given to the patient by the injection of antibodies which is taken from the animals or any other person, right? This is your natural passive immunity. So there are 4 types of acquired immunity. First one is natural active, artificial active, natural passive, artificial passive. I hope the difference is clear between these 4. Yes, clear to all of you? Okay. So now active immunity. Talk about active immunity. Active immunity is further of 2 types, naturally acquired or artificially active. So if I talk about naturally acquired active immunity, it develop automatically, or you can say it developed by an individual after from exposure to an infection, right? Active immunity occur when the body immune system respond to the antigen and start producing antibodies and lymphocytes. Naturally acquired active immunities follow illness or pathogen exposure, right? It never develop before the exposure of antigen. It always develop after exposure to the pathogen or antigen. Artificially acquired active immunity occur through vaccinations. Vaccines contain treated or altered microbes, toxins or part of microbes, right? A primary immune response, memory cells are formed. The person does not usually become ill. You have an idea about natural acquired active immunity? In the childhood, everyone get vaccinated. So at that time, we does not suffer with any disease, but the vaccination course is given to that child. Why? Can anyone answer for that? Why vaccination is given in the childhood? Why polio vaccine is has given? Because at that time child has not suffered with the polio. Then why we are vaccinated against polio? Because we gave treated or altered microbes to that individual, right? And we will establish their memory cells. So during their journey, if the person or the individual come in the contact with that type of similar antigen, their body start producing antibodies immediately, and the body of the individual is able to fight against that particular type of disease, right? So this is the artificially acquired active immunity. Types of vaccines. Yes, what are the different type of vaccines? First one is live attenuated vaccine. In this type of vaccines, we are using the microbes by weakened them. First, we weakened or altered the microbes, and then we multiply. Then we are altered their properties in this way that they can multiply only at low level, so that they are not producing or causing any harm to the individual, but they induce their immune system to produce antibody in the advance, right? So live attenuated vaccines is the one which contain live, or you can say viable microbes, but by having weakened multiplying system, their capability or ability is only to induce our immune system to be strong, right? Organisms can revert to the violent form and causes reverse. Yes, this is the you can say disadvantage of the live attenuated vaccines. Sometimes sometimes few of the organisms can revert to the violent form and it causes diseases. When it comes when a individual is getting exposure to an opportunity pathogens, right? In that case, the organism start revert to the violent form and start causing the disease to the individual instead of producing or making their immune system strong, right? Single dose vaccines can combine vaccine for different diseases. Vaccines using attenuated bacteria are difficult and not widely used. So this is the type of vaccine. This is the first type of vaccine, which is live attenuated vaccine, right? Now, inactivated vaccines. In this type of vaccines, we used killed pathogen. In the previous one, we are using the live bacteria, but we altered their multiplication capacity. But in this type, we killed the pathogen, which induce a weaker immune response, right? Booster shots are required to maintain immunity. They are safer than the attenuated vaccines because they cannot cause disease. Yes, the disadvantage of the live vaccines, we discussed that sometimes few organisms can cause diseases because they revert to the virulent form. But in inactivated vaccines, organisms are not able to producing diseases because we are using the killed pathogens, right? Then third one is toxicoid vaccines. Toxicoid vaccines basically contain inactivated toxins, which is your toxicoids. Since the product is inactivated, booster shots are required, prepared by incubating toxins with chemicals. What is the difference between these three types of vaccines? In live attenuated vaccines, single dose is required. But in inactivated or toxicoid vaccines, booster doses are required. And these two types of vaccines are safe because it cannot cause disease. The reason is the organism or the pathogens we are using is in the killed form, right? So to avoid multiple infection, vaccines are combined into single dose vaccines. So these are the three types of vaccines we discussed. Passive immunity. So passive immunity is basically the development of antibodies which enter from the body but from the outside. So passive immunity is the one which is passed through one to another. So it develops when antibodies start producing or enter into the body from the outsource. There are two types, naturally or artificially. Naturally is the one in which the antibodies passes from mother to fetus, and the artificial is the one in which antibodies are given externally to the individual, right? So first we talk about naturally acquired passive immunity. So it is also known as congenital immunity, right? It occurs when the immunity passes from the mother to fetus. And which type of immunity, or you can say antibodies passed? So IgG is passed from mother to fetus, and it is the second line of response. So it remain in the child 3 to 6 months after the delivery. Now, maternal antibodies also passes in newborns through milk, and milk is like two types. First milk, which is cholesterol, and breastfeeding. So the maternal antibodies is passed with both the types, right? Artificially acquired. It involves the injection of the antibodies with serum to the body. The serum can be used to prevent prophylactic reactions or treated therapeutic diseases. Anti-serum or convalescent serum we are using in this type of immunity. The immune system may recognize foreign serum protein as a non-self and mount allergic reactions. Immune complexes may form and serum sicknesses may develop. Right? In this scenario, sometime a patient is recognized as an antigen and start producing their own antibody against the antibodies. So this is the prophylactic reactions. Right? Now, serology. There are different serological tests or reactions which we are using to study the serum. So first of all, serology. Serology basically is the branch of immunology that deal with the serological reactions. It helps to diagnose microbial infection. Antigen antibody reactions are studied under this branch, right? Basically, immunology and serology come under the branch of microbiology. These are the part of microbiology. Here specifically we use the patient serum for our study. In the previous part we discussed about the immunity, vaccines, how the immunity of the patient develop. We discussed all the things, right? But in this specific part we need the patient serum to study about the antigen antibody reactions that run inside the body of the individual. So what is the function of serology? The function is to confirm the test, detect organisms in the tissue, aid MAD in following courses of the disease, medicines you are giving to the patient, right? And determine immune states. So these are the function of the serological reaction. Yes, the one point is like in this scenario, just go back to the immunity. See, if I talk about the natural immunity, no? So what was that? As I told you, the individual is come in the exposure of an antigen and start producing the antibody. How this process take place? So little bit I'm highlighting you that how our body start producing antibody. So how it is developed? First, the antigen enters into the human body, right? Apart from entering the antigen into the human body, just enter the human body, our immune system get evoked, it gets started. So first of all, T helper cell starts and it gives signals to the B helper cells and the T cytotoxic cells. So when the antigen is enter into the body, T helper cell activate and it gives signal to the B cells and your T cytotoxic cells, right? After that, B cells start producing plasma cells or the memory cells. On the other hand, T cytotoxic start producing your memory cells and T lymphocyte cells. Now, what will be the next cell? Plasma cells start producing memories and memory cell start memorizing the structure of that antigen. So in future, if reoccurrence of that organisms occur, they get start antibodies, right? And on the other cell, the cells produced by the T cytotoxic cell, it kills the antigen and again the memory cells start memorizing the structure of that organism. So in this scenario, in this similar way, our immune system work to produce antibodies, right? So this is the mechanism how our immune system work. And there are two type of immune system responses. There are two immune system responses. First one is humoral immune system, which is based on your B cells response, and the next one is cell-mediated immune system, which is based on the T cell response, right? I hope this thing is clear to all of you. So clear till now? If yes, so kindly give me heads up in the chat box. Then I will continue with that. Everything is clear to all of you? All right. So now the next is serological reaction. There are different type of serological reaction. First, starting with titration. So what is titration? Do you heard about the term titration? What is titration? Can anyone tell me? So let me know in the chat box if you know about that. What is titration? Okay, titration. See, are you able to see this picture? There are different test tubes, right? In the ratio, like 1:20, 1:40, 1:80, 1:160. This become double in every test tube. So this is the dilution of the antibody. And this dilution of the antibody is known as titration. So basically titration is the dilution of antigen or antibody solution to the most favorable conditions. So the titer is the most diluted concentration of the serum antibodies that react it antigen. A rise in titer ratio indicate disease. For an example, in this series, there are different test tubes, right? So if the agglutination is seen in 1:60, that means this is the visible antigen antibody reaction. But if you see in the test tube E and F, which is your 1:320 and 1:160, there is no agglutination. That means no visible reaction seen in this type of test tube. And this is the control one. There is no antibodies present. That is why there is no agglutination occur. So this is the titration. Titration basically means dilution of antigen or antibody, right? So if I talk about this, a person is having any type of disease, right? If you are using one part of the antigen and one part of the serum in this type of that, then when you transfer this, so it will become 1:40, and from this tube you will transform this next tube, and then next, and then next. Like this you transfer your serum into the next tubes. So your tubes will be diluted, or your antigen antibody solution will be diluted. This dilution of antigen or antibody solution in the favorable conditions is known as titration, right? So this is the titration reaction that we performed in the serological reactions, or you can say in the serology, for the study of antigen and antibody reaction. And high rise in the titer indicate diseases, correct? Now the next is neutralization. So what is neutralization? It involves basically antigen antibody reaction. So in this reaction, antigen and antibody get neutralized. How they get neutralized? Let's suppose I'm having one antigen and one antibody. Antigen and antibody combine each other and form a complex. So it becomes neutralized. There is no effect of antigen and there is no effect of antibody. This is known as neutralization. You can also understand with the example like positive and negative charge. If you give one positive charge and give one negative charge, it becomes neutral. There is no charge. In the similar way, number of antigen and number of antibody combine with each other and form neutralization. There is no effect of that particular antigen and antibodies at that time. So neutralization basically used to identify toxins, antitoxins, viruses and viral antibodies. It is as if a specific agent is suspected to determine if the toxin has been neutralized. A sample can be mixed with antitoxin, correct? Injected into the lab animal. Example, for an example, if you detect botulism toxin in food, in that case, what will you do? you can inject the toxin, right, into the lab animal and then add antitoxin, and then if you see any reaction, so that you can identify or you can see the result of that particular type of toxin. So these come into the neutralization reaction. Chick test is the example which we use to determine if a person is having diarrhea or is a person is immune to diphtheria, right? So this is a interdermal test. So chick test is the example of the neutralization reaction. So it is based on the principle of neutralization. So this is the second type of serological reaction in which we study about the reaction between antigen and antibody. So titration is used when you need to dilute the antibodies or antigen to see the titer of that titer, or you can say load of that organism in the body. And neutralization is used when you need to identify toxins or antitoxins. Correct? So clear with this? Is everything is clear till here? All right. All right. So moving toward next. Precipitation. So what is precipitation? Precipitation basically involve antigen antibody cross linkage in huge lattice. So in this image, see, this is an image. Are you able to see? See, this is the line of precipitation. Here antibodies present, here antigens are present. So this line is the line of precipitation, right? Or you can see in this dish, anti-A, anti-B, and this is the line of precipitation, right? So antigen A and anti-B is combining and form the line of precipitation. So you can understand one thing in this scenario. Whenever any antigen or antibody combine with each other, it form a complex, and it is visible in, and it is a visible form. So you can visualize in the form of agglutination, precipitation, neutralization, titration, any form. But whenever an antigen and antibody react with each other and form a complex, it becomes a visible reaction, right? So this is the example of the precipitation reaction. When anti-A, anti-B combine with each other, it form a line of precipitation, right? So basically precipitation is the involvement of antigen antibody and form a cross-linked huge lattice. In fluid, the molecule diffuses until they reach the ideal concentration. That means zone of equivalence. Let's suppose in this image there are 10 antibodies, right? And there are 20 antigens. Is the zone of, is they are equivalent to each other? They can neutralize each other? No, because the number of antigens are more as compared to antibodies. So there is zone whenever they meet the zone of equivalence, they form the visual reaction. So when they meet to the zone of equivalence, they form this reaction, and apart from them, the extra antigen or extra antibodies rest aside, right? So in immunodiffusion, antigen and antibody diffuses through a gel until they reach to the zone of electrophoresis, or you can say zone of equivalence. Outing tube technique and auxiliary plate technique we are using in the immunodiffusion techniques. When we are using immunodiffusion precipitation reaction, we can use tube technique as well as plate technique. And immunodiffusion technique is run by the using of gel. Correct? So this is your precipitation reaction. Now the next is immunoelectrophoresis. As the term is immuno, Immune system. Immune system is having antigen and antibody. Electro. Electro is your charge, electric current, and phoresis. Phoresis is the movement of the charged particle in the electric field, right? So in immunoelectrophoresis, diffusion is combined with the electrophoresis. So this is your plate, agar plate. One side is negatively charged and one side is your positively charged. Sample antigen is placed on the plate and then charge is introduced. Then through the solution antibodies start moving, right? And then precipitation line appear like this. This is your precipitation line. So these precipitation lines visible at the zone of equivalence where antigen and antibodies are equal in number. So are you aware with that? This is your antibody solution, right? And this is your antigen sample. Antigen and antibody start moving towards each other, and at the zone of equivalence, just a second. Yes. So this is your antigen and this is your antibody. Antigen is introduced in this and antibodies is also induced in this. Antigen and antibody start moving towards each other. After that, where the zone of equivalence meet, the precipitation lines you will be visible, right? So precipitation line will be visible at the zone of equivalence. So what is that? We will discuss in the detailed version. Gel electrophoresis. On a gel coated slide, this is your gel coated slide, an antigen sample is placed in the central wall. So this is your central wall and the antigen sample is placed. Now electric current is introduced and run to gel to separate the antigen by their electrical charge side. Unlike diagram, the separate antigen cannot be detected visually at that point, right? Now addition of the antibodies. These are your antibodies. So adding antibodies at that plate. Now these antibodies are already known. Known antibodies are added as antigen is unknown. Now diffusion of antigen and antibodies occur. That means antigen moves towards antibody and antibody moves toward antigen. Now there are precipitation line will be appeared after some time. These lines are seen where optimal concentration of antigen and antibody meet, which is your zone of equivalence, right? So this is your immunoelectrophoresis. So immunoelectrophoresis will occur at the zone of equivalence, right? Now the next, agglutination. What is it? Agglutination is an antibody interacted with antigen on the surface of a particular object causes object to clump together. The most common example of agglutination is your ABO grouping. ABO blood grouping is based on the principle of agglutination. Here, the antigen and antibody get interact with each other, but on the surface of a particular object, which causes object to clump together. Right? Visible reaction require less antigen or antibody if they clump together. In passive agglutination, antigen are absorbed onto a surface and antibodies are added. Agglutination is observed. Like in this image, you can see first antigen is absorbed onto the surface. Additionally, antibody is added, then agglutination is appeared. So this is your latex beads, bacterial antigen, antibodies attached together and they form agglutination, right? So this is the reaction of agglutination. So we discussed about titration. In the titration, there is a dilution of antigen and antibodies, right? Neutralization, in which we neutralize antigen antibody effect. Basically we used for the detection of toxins and antitoxins. Then precipitation is mainly or majorly performed in the liquid solutions, right? Agglutination. In agglutination, we see clumps, but we required an object for that, Right? Now the next, heme agglutination. Yes, this is the example of agglutination reaction, or you can say type of agglutination, heme agglutination or flocculation. So if I talk about heme agglutination, so heme agglutination is used to determine blood type. I already told you the example of agglutination is the ABO grouping. So this is similar to that. It detects viruses that cause agglutination of the red blood cells. Flocculation. Flocculation is come under precipitation and as well as agglutination. Here antigen exists in a non-cellular participate form that react with the antibody to yield large visual aggregates. Flocculation means floccules. Large aggregates appear on the surface of the slide, or you can say surface of the tube solution, right? So this is flocculation. So flocculation may occur in precipitation or in agglutination, right? So the difference is in flocculation, antigen exists in huge form and forming large aggregates, which is easily visualized to us, right? The complement fixation. Next reaction is complement fixation. So it can detect antibodies to the various pathogens. How it will work? So by this diagram, with the help of this diagram, we can understand this. So this is your test tube. This is having specific antigen, the patient serum, and the guinea pig complement are added to the test tube. So this is your antigen. This is your patient serum, which is containing antibody and also having complement of guinea pig. So now all the things are added in this tube, in which we incubate. After the incubation, patient's complement has been destroyed. So first of all, the three things added in the test tube A, and then it is incubated at 37 degrees Celsius for 90 minutes. Now your complement, patient serum and antigens are combined with each other. So the indicator system is added now, which is your sheep red cells. Correct? After the incubation of this tube, we are adding sheep red cells. So sheep red cells are act as an indicator. It consists sheep RBC and antibody that lies the RBC if complement is available. So further there are two cases. Case 1, which is your test tube C, and case 2, which is your test tube D. So first, in case 1, antigen and patient serum combine with each other and making a complement, right? So it does not combine with the sheep antibodies. So no lysis is occurred in the tube C. So if the patient serum contain antibodies to that particular type of antigen, a reaction will take place between the antigen and the antibody and complement. Because the complement has been used up, there is no lysis of sheep RBC will occur when the indicator system is added. Now in the case 2, there is no reaction occur in the patient serum, right? So patient serum is free now. Antibody is combined with the complement, right? So what is that? If specific antibody from the patient serum are not present in the serum, no reaction will be occur during the addition of the indicator and during the incubation. So whenever we add indicate, it combine and lysis the RBC. So it produce as a result lysis of RBC will occur. So this is your complement fixation. With the help of that, you can detect antibody to the variety of pathogens, right? So now come up with the next labeling method. Labeling method are also used in the serological reactions, in which we labeled over antigens, antibodies, enzyme with the different dyes with the enzymes with the different colors. So we are labeled antibodies, antigens with the fluorescent dyes, with the enzymes which help us in to identify different type of antigens. For an example, labeling methods are used to detect antigen antibody binding, right? So a fluorescent antibody technique can be used to detect antigen antibody binding by labeling antibodies with a fluorescent marker. So try to understand with this image. So this is your slide and this is your antigen. Antigen is of your syphilis spirochetes, right? Which is fixed to a slide. Now, patient serum is added to that fixed antigen, which we already fixed on the slide, right? Now, the patient serum is added. If the serum contain anti-syphilis, that means if the if the serum of that patient having antibody against syphilis antigen, so it will adhere to the antigen, right, which is present on the slide, which you already fixed. Now in the option C, this is your labeled antibody, which is the name of the method, labeling method. So this is your secondary antibody. This antibody is labeled with fluorescence labeled anti-human antibodies, which is added to that. Now, if the reaction is occurred, that means antigen and antibody combine to each other, it will go and adhere to that complement and forming a light or a colored mechanism, right? So that, yes, everything is okay. Everything is okay. All right. So continue with that. Yes. Just give me a second. There is some issue. Okay, so yes, continue with that. Okay, so we were at option D. So yes, adding fluorescence labeled dye in that complement. Now if the antigen and antibody connect with each other or combine with each other, then this fluorescent dye go and adhere to that particular complement, right? If there is no adherence, then this antibody will not attach to that particular type of complement. So in option D, if spirochetes are coated with anti-syphilis antibodies from the patient serum, the labeled antibody which you are using is react with them and producing a dye with a fluorescence, right? But on the other hand, if the serum has no antibodies, the patient serum is having no antibodies, no coating take place, the labeled antibody remain in the fluid and does not produce any dye or fluorescence. So basically, whenever you see any fluorescence, that means the combination, or you can say the complement of antigen antibody occurred, right? If the complement of antigen antibody does not occur, that means there is no reaction will be visible in the form of fluorescence or dye. What is the fluorescence? Basically, fluorescence is your colored object. You are able to see at the last of the reaction. As in the beginning of the serology, I told you about that whenever an antigen or antibody react with each other, it produces a visible reaction. It may be in the form of your clumps, in the form of precipitation, in the form of dye, in the form of color, in the form of titration, anything will be happen, but a visible reaction will be appear. This is the 100% sure, right? This is a 100% sure point. The visible reaction will be occur if the antigen or antibody combine with each other. Correct? So this is your labeling method. This is the method how you will label your antibodies, your antigen, to detect different type of antigen or their corresponding antibodies in the sample. Correct? So now next is RIA. RIA is a radio immunoassay. Basically, this is the extremely sensitive method. It is also used in the pregnancies, right, to detect any abnormalities in the fetus or any antibodies in the mother. So this RIA is used in the cases of pregnancy. Basically, as we discussed about the labeling method, so it come under that. So in this method, radio immunoassay, we labeled over antigen with the radioactive elements. So based on the competition between the radioactive labeled antigen and unlabeled antigen, the reactive sites on antibody molecules occur. Next is your radioallergosorbent test, which is used radioactive antiglobulin antibodies. Yes, in the image, this is your antigen to be suspected antibody bound to the plastic device, such as plastic plate with the cells. This is your plastic plate. So here antigens are coated. Now the test solution such as serum is added in which antibody is present. So here we are not sure about that antibody may be present or not be present because we are testing that serum. So if the antibody is present, it get attached to the plate on which antigen is already coated. But in case if antibody is not present, it will not attach. Now additionally, your labeling radioactive antiglobulin is added. If the antibody is attached, it go and attach to that antibody. So this is your primary antibody which is present in the patient serum, and this is your secondary antibody which we are using, the labeling antibody, right? So this is the way how radio immunoassay work or radioallergosorbent test work. Now the next one is ELISA. ELISA is your enzyme linked immunosorbent assay. It is similar to your RAST. It is basically used to detect the antigen and antibody in the patient sample. So in this reaction, like in the last, here we are using radioactive elements. But in this ELISA, we are using enzyme for labeling the secondary antibody. Most commonly, we are using horseradish peroxidase. This is the enzyme we are using in the ELISA technique. Basically, it is used to detect antibodies against HIV. So this is the process. Here is the HIV antigen. Now this is the HIV antibodies which is combined with the beads which are present on the HIV antigen. If no antibody HIV, so no antibodies, there are no antibodies, so there are no attachment occur. After that, enzymes are added, right? If antigen and antibodies connected, or you can say attached with each other after the incubation, then the enzyme attached to that particular type of complement, and then enzyme is added. You have to add substrate. After adding substrate, enzyme and substrate get combined and it form a colored reaction. So in this way, ELISA technique work, right? So this is your ELISA technique. Now in the labs, antibody recognize one epitopes. We are producing antibodies in our labs. As we discussed in the immunity, we are giving artificially antibodies to the patient which are prepared in our labs. It prepared by using lab animals. So in the labs, our antibodies are able to recognize one epitope. Epitope is the site which is present on the antigen, and paratope is the site which is present on the antibody. Now, which we are produce using myelomas. Myeloma cells are fused and activated B cells to form hybridoma. Hybridoma further producing desired monoclonal antibodies can be cloned. Monoclonal antibodies can be used in disease prevention, which we are giving to the patient or the individual, or immunodulation, which is controlling overactive inflammatory responses. So how we are preparing monoclonal antibodies? Basically, myeloma cells are fused and they activated B cells. B cells further producing your plasma cells, and plasma cells producing your antibodies, which I already told you about, that how our immune system works. Now the antibodies are produced, it will use as a disease prevention, or you can say controlling overactive inflammatory response. So this is the technique how we are producing antibodies in our labs, right? Gene probes, and gene probes are single stranded DNA segments. So basically DNA segments, PCR techniques. PCR is your polymerase chain reaction. Like we are produce antibodies, right? Now the antibodies are produced in a small amount and we require that type of antibody in a large quantity. So what can we do? We can amplify that antibody with the help of PCR technique, which come under gene probe or single stranded DNA segments. Here you can amplify the sequence of the DNA and produce a large amount of that segment, right? So PCR basically is a technique which is used to amplify the DNA strands. So your technique hunt complementary DNA fragments and emit signal. And gene probes and PCR used in HIV, HMP detection, water quality test. So this is your PCR technique first. This is your probes. You can denature it by increasing the temperature. After denaturation, there are two different strands. These two different strands act as a parent strand. Complementary to these strands, further two new strands are produced, right? And these cycles become continue and produces different and many segments. After the first cycle, we get 4 DNA fragments, and after the 20 to 30 cycle, this much amplified DNA sequences we are having, right? So this is all about your immunology and serology. We discussed all the things about the immunology, what is immunity, how our body produces immunity, and how we are producing antibodies in our labs. After that, what are the different serological reactions we are using to identify the diseases which is caused by the combination of antigen antibodies. So this is all about our immunology and serology. So I hope everything is clear to all of you. Now, if you have any doubt